Okay, our last talk is presented by Hera Hashma. The title of the talk is CRISPR-Cas9 mutagenesis of IMPAD in Xenophus laevis.
Sorry, I'm a little vertically challenged. Hello, my name is Hera, and today I have the honor of representing MCDB 4100 mutating Xenophus. The title of my project this semester was CRISPR-Cas9 mutagenesis of IMPAD1 in Xenophus laevis. A little bit of an overview of what we'll be going over. The goals of this course. So the primary goal was to understand gene function. It was to identify a gene of unknown function, and through mutagenesis, understanding the role it plays in the organism. A little bit about Xenophus laevis and why we chose that as a model organism, my gene of interest and the gene I chose, why I chose it, the methodology, which we mainly did CRISPR-Cas9 and in situ hybridization, the results, and future objectives. So first, Xenophus laevis. Why do we choose this as a model organism? Well, it undergoes rapid development, and I assume that most of us want to make a discovery in our lifetime. And compared to mice, it develops quite quickly, and it can withstand changes in environment such as temperature. So it's quite easy to maintain compared to mice. It's pretty inexpensive, and its embryos can tolerate extensive manipulations such as injections and dissections. So CRISPR-Cas9 involves injections, and we want to make sure that the embryos we're using, they don't die as a result of the injections themselves. The gene that I chose was inositol monophosphatase domain containing 1. Inositol is a sugar alcohol, and monophosphatase refers to the fact that the enzyme is a part of the IMPase family, and it's involved in the PI signaling pathway. So PI is a type of lipid in the plasma membrane, so it does affect a wide array of functions such as apoptosis, cellular signaling, information processing, and yeah, that's it. Existing literature. So the reason I chose IMPAD1 was because the Golgi apparatus is my favorite organelle. It's such a pretty organelle. And so when I looked it up on the OMIN database, when I sat down to find a gene, I searched the Golgi apparatus, and I found a paper in 2008 where researchers identified IMPAD1, and using immunofluorescence of human skin fibroblasts, identified that this was localized in the Golgi. So I was like, okay, great. Golgi apparatus, IMPAD1, I can do that. And then later, I wanted to make sure that the gene that I chose had some background information, that I wasn't going into the experiment completely blind. And I found that there was a case study of individuals in Turkey who had homozygous mutations in this gene and had chondrodysplasia, which is a sort of skeletal system abnormality. Later, I went on to Zenbase. Zenbase is everything Xenophus, so if you are a frog junkie, it's a great place to go. So I found some information on this gene. As you can see on your left, the x-axis represents the different stages of development, embryonic development, and on the y-axis, expression, transcripts per million. And you can see that the expression of my gene is most highly expressed at stage 9, or gastrulation, which is interesting because the skeletal system often doesn't start developing fully until stage 35. On the right, you can see adult expression of the gene, and at the top, actually, wait, can I use a pointer? Ooh, okay. At the top, LNS refers to long and short, so Xenophus actually has 2 copies of each chromosome, long and short. But this experiment did target both copies of the chromosome. But you can see it's most highly expressed in the brain and in the ovary. So in order to better understand gene function, we used CRISPR-Cas9 technology, which many of you probably have heard of, and it was a lot of fun. This was my first time working with it. This is my gene here, IMPAD1. It's about 17,000 base pairs long. I chose the exon, chose the largest exon to work with. And the way that CRISPR-Cas9 generally works is you identify a target sequence of interest, and you design a guide RNA, which will recognize that sequence. Cas9 is an enzyme that will then go in and bind to that region and cut both strands of the DNA. After that, the cell will try to repair the cut, but it is an error-prone mechanism, and it will induce a mutation. And this is what we wanted. Bit of an overview of our experiment over the course of the semester. I won't go into detail because we are on a time crunch, but if you're interested, feel free to come back by our mutating Xenophus posters, which are over here. But we did design the oligos, so we used the software, Ape software, to find a sequence in our gene of interest that would fit the criteria. We then designed our oligos, we transformed bacteria with plasmids containing that sequence. We then confirmed that those bacteria had picked up the plasmid, and then we determined whether that sequence had been ligated properly, and finally injected Xenophus laevis embryos with those guide RNAs in Cas9. Our results, in a nutshell, death, sudden death. So we did this twice. We carried out this experiment twice. The first time we did this, each person injected about 10 embryos, 10 to 15 embryos. They all died within a few hours, including the uninjected embryos, which served as our negative control. Second time around, out of hundreds of embryos, only 7 uninjected negative controls survived. 4 tyrosinase guide RNA and Cas9 embryos, which served as our positive control, and then 1 MYL4 injected embryo, which was a classmate's experimental embryo. Shout out to Charles. I don't know what you did right, but it worked. But what this sort of implies is that the experimental failure wasn't a result of the technology not working or even my gene causing that phenotype, but rather it was a problem with the embryos, that they were probably low quality because the negative controls died as well. Another way to analyze gene expression profiles is through whole mount in situ hybridization. So in order to determine and understand gene expression at different points in development, like that graph I showed you before, this is a really nice way of localizing detected in specific sequences at various stages in spatiotemporal gene stages. Okay. So the way it's done is we design RNA probes that can then, I'll just keep going. There's a really nice schematic here, like the other ones, so just imagine that's there. So we design RNA probes that can anneal to our sequence of interest, and then it was tagged with DIG. When antibodies contact that, it will cause a colorimetric reaction, and then it can be visualized under different types of light. These are the results, so I really hope it shows. So these are embryos, Xenophus laevis embryos at different stages of development. On your left is the positive control. As you can see, there is a purple band on the top right embryo in the mesoderm, in the middle of the negative control with nothing, which is good because that's what we want. And then my gene in pad 1. So it looks very close to the negative control with the exception of the embryo at the top right. I don't know if you can see it in the projector, but there's a very light purple band across that embryo in the mesoderm, which is what I expected because the mesoderm is responsible for the formation of the skeletal system. Future objectives. Since the Cas9 experiment didn't go too well, I would definitely want to repeat it in the future, and then do a genome-wide assay to confirm if those embryos had actually if the cells in that embryo had been affected with the mutation. And then also due to the animal protocol here, the animals are not allowed to develop past stage 35 of development, which is the point at which they can feel pain. However, some literature has suggested that because IMPAD1 affects skeletogenesis, that phenotype may not come until later stages in development. So if I were able to do this experiment, I would definitely want to play with the idea of how, when that phenotype arises, and if it's at later stages of development, and even in adults. Thank you so much for listening to me. I would want to acknowledge the funding for this project that was provided by the Chancellor's Award and the Europe Team Grant to Dr. Bilge Bursoy and Dr. Michael Klimkowski, and the course fees paid by the students in the MCDB department. And thank you so much to Dr. Tyler Square and Dr. Dan Medeiros for providing some of the materials that we used in this lab. Thank you so much. Any questions? I'll try to answer them, but I still don't know what I did, so yes. Yes. Okay, yeah, so a xenopus is a pseudotetraploid, so there's a long evolutionary history involving the fact that it has like 2 copies of each chromosome. So in the beginning, when we picked our gene, we did confirm, or for my gene at least, I confirmed that it was present in both the long and the short chromosome. So when we carried out the experimental procedure, it did hit 2. I know some of my classmates did have genes that were only present on one or the other, and so they carried out their experiments that way. Yes? No clue. If you want, you can come by our posters, and Dr. Versoy, I think she's in the audience, she's the one who carried out a lot of the experiments, so she could probably answer that question if you want to do that now or later. They were in the same stages, so we went in one day and Dr. Versoy actually, she mashed up the testes of the male frog and then she fertilized the eggs, and so they were all fertilized at the same time, and then we waited about half an hour and then we all injected. So there were, I think we only had like 3 machines in order to do it, but there was only like a 5 to 10 minute difference between the injection periods. Any other questions? If you get a chance, do take this lab. By far one of the funnest labs I've ever done. Thank you so much.